Hi guys, and welcome back. Today we are looking at the structure of DNA. And the structure of DNA starts by looking at the structure of the monomer of DNA, which is the nucleotide. So DNA has 4 separate nucleotides that all have some things in common. Every nucleotide is attached to a phosphate group, and the phosphate group is often represented as a small circle. The small circle, when it gets linked together, may have oxygens showing on either side of it, but phosphate is typically this little circle. The sugar, in this case deoxyribose, is going to be a pentose sugar. So it is making a pentagon or a little house, a little pentose sugar here that connects to the phosphate, making the backbone. And those parts are the same for every nucleotide. But each nucleotide has its own nitrogen-containing base, and there are 4 nitrogen-containing bases: adenine, guanine, cytosine, and thymine. Thymine gets replaced by uracil when we get to RNA, which is something we'll have to deal with next chapter. But for now, A and T, G and C, adenine and guanine, cytosine, and thymine. And each of these attaches to a sugar and phosphate backbone, which we see here. The nucleotides attach to the sugar phosphate backbone and are eventually going to pair A to T and G to C according to Chargaff's rules. And so Chargaff was a researcher who found that in every kind of DNA that you could identify, the proportion of A to T and G to C was proportional. And so from that, Watson and Crick derived that that probably meant that A was combining with T and G was pairing with C when the molecule formed. A and G are known as purines, and they are double ringed. So this purple one here is a purine. This nitrogenous base over here is also a purine. T and C, thymine and cytosine, are pyrimidines, and they have just a single hexagon shape. So they don't have this pentose sugar, they just have this hexagon shape here. And one way that they told you in the other video to remember the pyrimidines was that cytosine and thymine have Y's in them, which is helpful if you remember their names. One of the things I find to be useful, one thing that I remember is pure agony. Pure agony tells us that the purines are A and G. And it even goes one step further, which is going to help you in today's activity, because A and G would be pure agony to draw. They are the bigger, more complicated, double-ringed structures. So that's a way to remember that they are the purines and that the purines have that double-ringed structure. Watson and Crick took that foundation of knowledge, the structure of the nucleotide, the Chargaff's rules that paired A with T and G with C, the concept of the sugar and phosphate backbone, and Watson and Crick used the photo 51 that Rosalind Franklin had taken of the structure of DNA and realized that it was a helix. Even more so, they realized that it was a double helix, which means that these two strands, run what we call anti-parallel. These 2 pens are parallel. I have put the caps at the top. But if I were to flip one of the pens over, these are now considered anti-parallel. What that means is it's like lanes on a freeway or lanes on a street. They are parallel to one another. They're not going to intersect, but they're not going in the same direction. They're going in opposite directions. And so what we see about DNA is one strand runs up while the other strand runs down. And now DNA is often to the side or at an angle or straight up and down. And so instead of saying up and down, we use a terminology called 5' and 3'. I'm not going to ask you specifically about what those numbers mean, but they indicate whether the phosphate is on the top or the phosphate is on the bottom, and so we would call that the 5' end or the 3' end. Once those two strands run anti-parallel, they hydrogen bond in between them. And the hydrogen bond is so important because it is a strong, stable bond, but it's not a permanent bond, like a zipper. Okay, zippers are amazing. Zippers hold your sweatshirt together. They hold your jacket together exactly as strong as you need it to when you're out in the cold, when you're snowboarding. You want that jacket held together. But when it's time to take off the jacket, you can just easily unzip it and take the jacket off. You're not stuck in this tight jacket and you can't get out and you're uncomfortable. It holds fast when it needs to, but it is easily undone. And that is the beauty of the hydrogen bond in DNA as well. It holds these two strands paired together, brilliantly strong. But when it is time to replicate the DNA or to transcribe the DNA into proteins, the hydrogen bonds can very easily be manipulated to just break apart from one another and allow in the enzymes that are needed to do the job. So C and G have 3 hydrogen bonds between them, and that rhymes, CG3. And that's how you're going to remember that when you look at a structure of DNA like this, the ones that have 3 phosphates between them are cytosine and guanine. You'll know that guanine is the double ring structure because pure agony tells you that A and G are the double ring structures. A, T have 2. So if GC3, that all rhymes, have 3, then A and T have 2 hydrogen bonds between them. And once again, you would know that adenine was the double ring structure because of that pure agony memory trick that we're going to use as well. So if you can remember pure agony and GC3, then you would be able to look at a fully unlabeled diagram of DNA, and you would be able to determine where A, T, G, and C were placed within that structure. And you will need to be able to do that for today's activity and for the test. All right, you guys, that finishes up our structure. You have a worksheet to complete before I see you in class tomorrow.